Hello there. Good afternoon, or good morning, good afternoon, and good evening, wherever you're joining us from today. My name is Inish O-Doherty, and I'm the Vice President for Immunology and the Hematology Program at the Critical Path Institute. I also run the Type 1 Diabetes projects, along with our team internally, as well as the Transplant Therapeutics Consortium. So it's my great honor to be able to welcome you all here today to the first day of what is going to be a two-day workshop titled The Design of Clinical Trials in New-Onset T1D, Regulatory Considerations for Drug Development. So as part of my welcome, I'm going to give you a little bit of background on who we are at C-Path, the Critical Path Institute, and then be able to jump into how the 2 days are going to play from an agenda side of things, then briefly touch on a couple of housekeeping items, and then pass it over to folks who are going to be giving introductory remarks. So as I said, just to give a little bit of a background and explain who we are at C-Path. So the Critical Path Institute was formed in 2005 in response to the Critical Path Initiative, which was a publication put forward by FDA outlining areas of unmet need in the drug development process, but also areas in which solutions or tools could be brought forward to address those unmet needs. So we are a 501c3 not-for-profit. We're based in Tucson, Arizona, and we also have an office in Dublin, Ireland. And since 2005, we've had a memorandum of understanding with C-Path. So the quote there is taken directly from the Critical Path Initiative report, and it speaks to the idea that there's a need to foster the development of new evaluation tools to inform medical product development. And that's really what we do with tools being a broad encompassing statement. So how we do that at C-Path, just to give a little bit of a greater detail of who we are, is that we act as a trusted neutral third party. So again, as I said, we are a 501c3 not-for-profit. And what that enables us to be able to do is to convene settings like this, or in other aspects, public-private partnerships. But most importantly, that the convening of these entities happens in the pre-competitive space. And by virtue of that, we're able to bring together folks from different pharmaceutical organizations, patient advocacy groups, academia, clinical researchers, and government agencies for the purpose of sharing data and expertise. And because this is a neutral pre-competitive ground, we do not seek to forward conversations about an individual asset, an individual company's interests, or an individual development program. Rather, we focus on the intrinsic unmet needs within a specific indication or therapeutic area. So we convene these neutral pre-competitive grounds, typically through the construct, but not in an absolute sense, through consortia. And on the bottom right side of the slide is just a snapshot of how these consortia look like, again, with folks from different industry organizations, the health authorities, patient advocacy groups, and government organizations, along with clinical researchers. So we convene this neutral space, we bring our core competencies to bear, which are regulatory science, data science, quantitative medicine, and the development of clinical outcome assessments to be able to put forward solutions for specific unmet needs. So again, the virtue of this pre-competitive space isn't that it's just a one-time conversation, because frequently these unmet needs and the proposed solutions are rather challenging, and they take time and investment and the attention of lots of different bodies. So by having this conduit to communicate with the health authorities in an iterative fashion, we can seek to develop new tools, methods, or solutions that can help accelerate the assessment of safety and efficacy of medical products. So what does C-Path look like? As I said, we take the form of consortia, public-private partnerships, or programs. In 2021, we have 21 different public-private partnerships. And again, there isn't a test on this at the end, don't worry. Simply, this is just to show the cross-section of coverage that we have with positions in the neuroscience space, rare diseases, but also in specific indications like type 1 diabetes and the Transplant Therapeutics Consortium. Some of our consortia are a little different because drug development is hard and needs different solutions from different contexts. So for example, the Predictive Safety Testing Consortium is therapeutic area agnostic, but seeks to develop solutions in the form of safety biomarkers to be able to advance drug development. Also, we have consortia like the PRO Consortium that seek to be able to develop solutions in the form of patient-reported outcome measures for specific indications. So they cut across a variety of different indications. But although a lot of the consortia, the public-private partnerships, are very different in C-Path, the one unifying piece among them is the fact that they are all brought together in this pre-competitive space to be able to identify specific unmet needs in indications, and then be able to bring the appropriate constituents together to talk about those unmet needs and propose rational, data-driven solutions. And really, the workshop here today is a bit of an embodiment of that as well. So enough about c-path and on to where the focus of the workshop is going to be today. So we're going to be talking about new-onset T1D, and I borrowed this figure from the publication listed below to show the area in which we're going to be looking at. So new-onset T1D is, as what this slide puts forward, the stage 3. So once you've met your clinical diagnosis. Now, that is not to say that there are not unmet needs preceding stage 3 or clinical diagnosis. It's merely to say that the purpose of this workshop, we're going to be focusing on new-onset T1D or stage 3. I also borrowed this figure because it highlights a couple of the biomarkers and measures that are pertinent and will feature in today's discussion, like c-peptide, and also different measures of glycemic control and insulin dose and resistance. So knowing that we're going to be focusing this conversation over today and tomorrow on this stage 3 new-onset T1D construct, who's been participating, and to be able to give you, the audience, a little bit of background about where this workshop comes from. So the planning committee has helped form the agenda, select the speakers, and be able to put together the questions, which have helped guide the individual presenters and put forward questions about what are the unmet needs in new-onset T1D. So you'll see we have folks from c-path, my colleagues, but we've had fantastic representation from the European Medicines Agency, EMA, on the left-hand side, and also the US Food and Drug Administration on the right side. We've been very fortunate also to have representation from Inodia, the EMA consortium in Europe, but also TrialNet within the United States. Very importantly, we've been able to incorporate patient voice through JDRF in this process too, but just to give a resounding thank you for all the time, attention, and focus that this committee has put in, spanning time zones from the West Coast of the United States to Central Europe. So how has this planning committee come together to be able to build an agenda, again, which is focused in pre-competitive dialogue that I'll keep on saying on the unmet needs and what potential solutions might be brought forward that are rational and data-driven. So we're basing this in a 2-day workshop, as you well know. Each day is about 4 hours long. The first session of day 1 is going to be hearing from the FDA and EMA on their perspective on the regulatory framework for new-onset T1D. From there, we'll be transitioning into session 2, looking at the scientific framework for the rationale of c-peptide preservation and its use as a clinical trial endpoint in T1D. For this, we're very fortunate to have a number of presenters and clinical researchers, which are going to speak to the natural history of c-peptide and its relationship to the progression of type 1 diabetes. We're going to hear about islet transplantation and the reintroduction of functional beta cell mass, and subsequently c-peptide and its impact on clinically meaningful outcomes. We're going to touch on the heterogeneity encapsulated within this chronic disease and ways in which we might be able to understand that for the purposes of clinical trials. And then we're going to hear from an industry perspective. So on the session 2, we're going to have a panel session after that, and then we'll close out day 1. When we reconvene in day 2, we're going to be opening up the conversation on what we've titled Establishing and Confirming Clinical Benefit. So c-peptide is a biomarker, and when we want to use biomarkers in the context, particularly of registration studies, we need to be mindful that we are indeed making benefits in clinical meaningful outcomes. So that term can mean different things to different constituents in the drug development process, but it does mean very definitive things to the folks at the health authorities. So being able to understand the relative perspectives from the health authorities, clinical researchers, is critically important to be able to know where we are and where we might go forward together. We're also very fortunate to be able to have patient perspective and caregiver perspective in this session, with a fantastic talk as well there. There will be a session there will be a panel session after session 3, and then we'll move into session 4, which seeks to be able to take the themes that have been discussed through the previous 3 sessions with 3 additional talks to consider the issues on overall study design. So bringing together the different elements of using c-peptide as a primary endpoint, as an endpoint in clinical trials, along with the other measures for confirming clinical benefit. The very tangible output of the workshop is going to be that the recordings will be made publicly available so that people can go back and look at them afterwards. Also, the meeting proceedings will be published with input from presenters and panelists. The slightly less tangible but really critically important part of all these conversations today is that this is an ability to communicate in the pre-competitive space. We are not going to solve every unmet need in new-onset T1D clinical trial design today or tomorrow, but we can progress the conversation and understand where we're coming from as different constituents and parties, be solution focused, but also grounded in science and what data we can put forward. And I think if we can bear that in mind during the panel sessions and the questions that are asked, and I definitely ask presenters to ask questions, we can move this dialogue forward. So I'd be remiss if I didn't thank all the folks from C-Path who've been working in the back of house here for the last couple of months to pull this off. And I really am extraordinarily proud to be able to call them teammates, and the fact they've done so much work in the background. So just briefly touch on some housekeeping items. This workshop is being recorded, and as I mentioned, we will make the talks available at the end. Attendees will not be able to share their video or speaking capabilities because we have so many folks dialed in today. But what we'll ask folks to do as they're watching talks, as they're listening to panels, and they want to ask questions, is to use the Q&A tab. We'll do our best to be able to go through these questions. We've got a lot of attendees, but we will seek to be able to get to as many as we can. Also, if you're having technology issues, please also use this Q&A tab. So finally, where are we going next? We're going to be hearing some introductory remarks from Dr. Irony at FDA, followed by Dr. Kowalski from JDRF, where he'll be speaking about the unmet need for disease-modifying therapies in new-onset T1D. From there, we'll be rolling straight into session 1, getting the perspectives of FDA and EMA with regard to the regulatory frameworks for clinical investigations for new-onset T1D in their own geographic remits. So from there, after that, we'll take a break and then come back into session 2. So I wanted to be able to thank you all again for making the time to participate today. We've had a great number of attendees who've logged in. And again, stay participated, stay involved, ask questions, and we'll do our best to get through as many as possible. And I'll see you again at the end of the day. Thank you so much.
Hi, good morning and welcome to this important workshop on regulatory considerations for drug development in new-onset type 1 diabetes. My name is Ilan Irony. I'm the Deputy Director of the Division of Clinical Evaluation, Pharmacology, Toxicology in the Office of Tissues and Advanced Therapies in CBER, FDA. I especially want to thank the Critical Path Institute for their effort into organizing the workshop and the logistics in putting together this endeavor. Over 100 years ago, a major milestone occurred with the discovery of insulin as a treatment for diabetes. That discovery transformed type 1 diabetes from an almost uniformly fatal condition into a chronic treatable disease. Despite the tremendous progress made since then with the use of new insulin analogs and devices that monitor glucose continuously and control rates of insulin delivery, there is still an unmet need. Hypoglycemia remains a considerable challenge in meeting glycemic goals, and chronic microvascular as well as macrovascular complications continue to afflict our diabetic patients. Type 1 diabetes is not a uniform disease. Although all patients share a common pathogenesis, the rate of beta cell reserve at the time of diagnosis and the rate of decline in that reserve, as measured by circulating C-peptide, vary significantly among individuals. Therefore, there is an opportunity to intervene in the disease course and a hope that even if the autoimmune destructive process cannot be stopped completely, that can be attenuated or delayed. In this workshop, experts in immunology and endocrinology, in ethics, and drug clinical development will be presenting their views to different stakeholders: industry, regulators, academia, patients, caregivers, and advocacy groups, focusing on the challenges and opportunities presented to us by the state of science and knowledge of the disease in 2021. In the next 2 days, you'll hear many fascinating presentations and discussions. On day 1, there will be 2 sessions, as Inish pointed out. The first will be about the FDA and EMA perspectives and tools to regulate and oversee drug development, and the framework for trials in new-onset type 1 diabetes. The second session will focus on the scientific basis of assessing beta cell reserve decline and the role of C-peptide in the clinical development, from a biomarker to be tracked to a surrogate endpoint to show efficacy. On day 2, in the first session, we'll hear various perspectives into the design of drug trials for new-onset type 1 diabetes, with a goal to confirm the benefit of these interventions. We'll hear from patients and caregivers, from regulatory agencies, and from industry. On the second session of day 2, we'll hear specifically about issues related to the design and feasibility of such trials. ranging from ethical, clinical, and statistical considerations. After listening to the important and interesting presentations, it's important to have a discussion among the different stakeholders. The panel discussions will then attempt to create common understanding of the different perspectives. They will also bring more clarity on the path to have products that halt the currently inevitable destruction of beta cells, and I personally hope that in the future, these drugs will allow us to delay or prevent the disease altogether. Thank you.
Hello, I'm Dr. Aaron Kowalski, JDRF CEO, and I want to thank the FDA and the EMA and C-Path, all the presenters and moderators for participating in this very important discussion with the Critical Path Institute. My talk is the patient perspective and unmet need in T1D. Now, a little bit about JDRF. We have fought to advance T1D research since our founding 50 years ago and won't stop until we find a cure for T1D. Our vision is a world without T1D. And our mission is improving lives today and tomorrow by accelerating life-changing breakthroughs to cure, prevent, and better treat T1D and its complications. And we've fought type 1 diabetes through science, driving research to advance treatments to cure type 1 diabetes and improve lives. And we've seen significant progress. We've relentlessly pushed forward, bringing in through collaboration researchers from around the world and have seen significant results. I'm proud of the work that JDRF did in the world of artificial pancreas research, accelerating the development of the first generation systems by at least 5 years, work on continuous glucose monitors, advancing beta cell regeneration, and other drugs and therapeutic approaches to delay the onset of type 1 diabetes. In this slide, you'll see our focus on disease-modifying therapies. Type 1 diabetes is an autoimmune disease, but unfortunately, it's one of the only autoimmune diseases that does not have therapies that actually modify the disease itself. Its incidence is increasing and globally, and it's present in all populations globally. We know the natural history of type 1 diabetes progression. It's been well characterized, often compared to other autoimmune diseases. But again, despite the advancement in our understanding of the disease and the research that's taken place, it's one of only a handful of autoimmune diseases where there aren't disease-modifying therapies approved. This graph coming from Insula et al. Shows the natural history of the development of the disease in 3 stages, where we know the autoimmunity is present before the onset of hyperglycemia. And this gives us an opportunity, we believe, to intervene before full stage 3 onset type 1 diabetes. This is patient registry data that shows, and I think I often say it's a myth buster, that insulin alone can treat type 1 diabetes. And we know that even in 2021, that managing type 1 diabetes is still very, very difficult. This data from the T1D exchange shows that the bulk of people do not achieve type 1 diabetes glycemic goals as measured by the A1C. And this data is from a population that's enriched for people who go to diabetes clinics. It's likely that if we had better data in the United States, that these outcomes would be even worse. And it highlights a tremendous unmet need. The punchline of this slide: 4 in 5 children do not meet A1C goals, and 2 in 3 adults do not meet A1C goals. Even with the advanced technologies that we have in 2021, we need to do better. I think we also need to highlight the burden of type 1 diabetes, and I'd be remiss if I didn't reflect upon very compelling testimony at the recent teplizumab AdCom meeting, which from a broad representation of people with type 1 diabetes, myself included, patients, caregivers, clinicians, researchers, spoke about the burden of the disease and the unmet need that exists in type 1. The T1D is not just glycemia. It's not just insulin injections. It's a combination of factors: food, exercise, stress, blood sugar management, insulin management, and the list goes on and on. And yet, as we showed in the last slide, outcomes are still suboptimal. The message is crystal clear. We need to change the course of this disease and improve diabetes outcomes. We know diabetes leads to poor outcomes. Complications such as kidney disease, retinopathy and diabetic eye disease, cardiovascular disease, and these are due to increased disease duration. Longer time spent hyperglycemic increases the risk for these terrible complications. We need to do better. We need disease modification. So we need disease modification urgently. Insulin has been around for 100 years, but it is not a cure. Tremendous advancements have happened. The state of diabetes care today shows significant improvements, but still shows the opportunity to drive much further. Let's be honest. As a scientist, it's crystal clear that subcutaneous insulin is not physiologic. The body did not intend insulin to come from the periphery. It intended it to flow into the portal vein and through the liver, and thus maintaining glucose homeostasis with sub-Q insulin is virtually impossible. We know that from the data, and leads to hypoglycemia risk in the short term and a hyperglycemic driven long-term complications. And that is driving poor outcomes. So we need a paradigm shift here. We need new ways to treat type 1 diabetes that address the underlying pathophysiology, not just treatments, but disease modification, so that we can give people with type 1 diabetes and their families better outcomes. This is going to take collaboration, disease modification. I'm proud of the role JDRF plays, but it's not just JDRF. It's a variety of stakeholders. We are at an inflection point here. And in the next 2 days, we're going to hear about what we know about C-peptide, a very strong biomarker for beta cell function, and data supporting its use as a primary endpoint in New-Onset studies. You're going to hear the voice of the patient and caregivers during tomorrow's patient roundtable. And I would urge you to listen carefully to what's clinically meaningful to them and their families. It's not always just A1C, and that's going to be very clear as you listen. We'll discuss clinical outcomes that are appropriate to support the use of C-peptide and talk about study design. What's possible? What's feasible? How to think about this unique patient population adjusting to an overwhelming and life-changing disease. So the group here today, regulators, FDA, EMA, industry, researchers, academia, funders, patient caregivers, we all need to link arms. We all need to work together. We have an opportunity to address an unmet need that still exists in type 1 diabetes and change the course of the disease, ease disease management, and most importantly, thinking about bringing cures to people with type 1 diabetes. We're going to need to work together. None of us can do this alone. And I can't thank, again, the Critical Path Institute for putting together this incredible opportunity to share these perspectives and get at this amazing opportunity. Thank you.